Today I am briefly talking about electrophoresis. As a brief overview, we have amplified our gene of interest in these two samples by doing PCR, and today we will see either that specific gene is mutant or wild type in these two samples which are coming from two different animals. And for this, first of all, I have to cast a gel. I will take 2 grams of agarose and then I will put it in 1x TB buffer. The volume of the buffer will be like 100 ml, and then I will microwave it for 2 minutes so that the agarose is dissolved in the buffer. After that, I will take out the gel and then I will put 1.5 microliter of ethidium bromide. Ethidium bromide is quite carcinogenic. That is why we have to be quite cautious while using ethidium bromide. And after that, I will put this gel, agarose gel, into the gel container to set the gel. And I will use these combs for creating a space for loading my DNA. These bigger combs are used usually for creating a space up to 50 or 60 microliter, which is in case of cloning. But for doing genotyping, we use quite smaller comb so that we load 20 microliter or 15 to 20 microliter of our product. And we will allow to set the gel for 30 minutes. After 30 minutes, we will get our gel. And after that, we will put the gel in the gel electrophoresis container, and we will fill the container with 1x TB buffer, and then we will load the DNA ladder here. And this DNA ladder is an indicator for matching our product size with a standard. And after that, we will load our sample 1 and sample 2, sometimes many samples in this each comb, and then we will connect this electrophoresis chamber with the power supply. Simple rule of thumb is the red goes to red and black goes to black. It is always from minus to plus, the running is. And we will set the volt up to 100 and the time for 70 minutes. Now the percentage of the gel we are using, the time we are running the product, and the volume, the voltage we are using, all these depends on the size of the product. For example, in my case, the product size is something between 400 and 500. For that, I am using these conditions because, for example, if the product size is 100 base pair to 1000 base pair, we will use 2% gel. If the product size is more than 1000 and less than 3000, we will use 1% agarose gel. And if the product size is more than 3000, we will use something like 0.5% of agarose gel. In the same way, if the product size is smaller, we will run it for short time. And if the product size is longer, we will run it for more time. When I say product size, it means the implicon size. In other words, it is the size of the gene we have amplified. It is the size of the gene between the start of forward primers to the end of the reverse primers. For example, if I have put my forward primers are binding at the start of the gene, and the reverse primers are binding something like 500 after the first forward primer nucleotide. It means only that area between the forward and reverse primer will be amplified. And for example, I have a gene, the total base pairs in the genes are 50,000 base pairs. And if I will put my forward primers at the start and reverse primers at the end, then the size of the product will be like whole of the gene. It will be like 56,000. So it depends on the person who are designing the forward and reverse primers to get a specified product. And for example, I have designed my primers in a way that my mutant product is 500 and wild type product is 400. And in this way, we can design our experiment. So I will run it shortly for 70 minutes, and then I will take this gel out, and I will go to the imaging system. I will put the gel here and I will scan it, and it will give me a nice scan. These are the ladder 100, 200, 300, 400 and 500, 600 base pairs, and this is the standard by which we are matching our product size. And here I had loaded my negative control to see that any unspecific band should not appear there. So there is no unspecific band. That is why it indicates that these two bands are true. And now this product size is like coming in 500, and my wild type is like 400 and mutant type is 500, for example. So if my mutant is like 500, this will be the mutant animal, and if my wild type product is like 400, this will be the wild type animal. In this way, I will be able to indicate it that either the animal is mutant or wild type. For example, I had designed my implicon size for the mutant like 500, so it is coming in 500. Now the animal is mutant, and if I have designed the wild type product size as a 400, now this animal is a wild type animal. So in this way, we can use the PCR and electrophoresis to know that either the animal is mutant or wild type. So that was one of the example where we can use PCR for any scientific purpose. For example, in case of genotyping. But PCR may be used for other purposes. For example, if we are doing gene expression analysis, we will take our RNA out and we will do reverse transcription of this RNA in the PCR by adding some reverse transcriptase. This is another application of PCR, and then after that we may amplify this copy DNA, cDNA. So this is another application of PCR. Another application of PCR is, for example, if we are doing cloning, we can take a specific sequence of DNA, we will amplify this DNA, and we will cut it with restriction endonucleases, and we will put it here in this plasmid, and we will clone it. So PCR is also used during cloning purpose. We can also use PCR during mutagenesis. It is a very cool, interesting technique. For example, we have a hypothesis. We say that this protein A is targeted by enzyme B in a specific, for example, lysine nucleotide. In other way, for example, we have protein A. This protein A, our hypothesis is that it has a lysine amino acid, and this enzyme B is attacking on the lysine of the protein A. So to test this hypothesis, what we can do is we can do mutagenesis. We can take this protein A gene out, and we will mutate this amino acid. For example, we will mutate this lysine by putting another amino acid, another nucleotide here into, for example, like for example, like in another amino acid like methionine. And then we can see that when we will mutate this lysine, then we will see either this enzyme B is attacking the protein A, are not. So if it is still attacking, it means our hypothesis is not working. If now the enzyme is unable to attack the protein A, it means that the only site where the enzyme is attacking on the protein is that lysine residue. So that technique is called mutagenesis, and for mutagenesis we can use our PCR. And also during sequencing process, next generation sequencing, second, third generation sequencing processes, when we are making our DNA library, in that process we can use PCR. So, and even in the methylation studies we can use PCR. So the idea of using PCR for genotyping was how the PCR works and how we do genotyping, but the PCR has many other applications, and it is a gold standard technique for DNA studies. And so that was something about DNA. In next topic, maybe I will pick RNA, how we can isolate RNA and what type of analysis we can do with RNA. Thank you so much.